All right, let's talk a little bit about chapter four. Chapter four is going to be primarily dominated by DNA, DNA synthesis, how cells grow, how they replicate and then divide, materials that are a little bit more genetic focused. So let's discuss number one, just what genes are and what do they represent? And again, like I mentioned in chapter three, how does chromosomes, DNA, everything kind of relate to each other? So, introduction. So we're gonna need to know a little bit about some of this DNA genes and how they affect us, because I mean, obviously there's so many diseases that we know that are inherited in some way or another. There are some diseases that are specifically inherited, whether it's passed on mother to son, or if you have, maybe a family member that are carriers of the actual disease. And then you have another, the husband or the wife is also a carrier. We know that if they're both carriers that of this, certain illness, maybe like sickle cell anemia, then that actual disease, if both parents are carriers, we have a rough understanding, very specific understanding of what are the chances that if we have two carriers of sickle cell anemia, what are the chances of a baby with the full disease? So we'll kind of discuss that. And if both parents are carriers, there's a 25% chance of a baby with the full disease, even though both parents are just carriers. They don't have the disease. Now, if both parents have the disease, then obviously the kids will have the disease. 100% a chance, 100% of the time. Uh, we call that kind of genetic inheritance, where we can make a very, very specific determination of the outcome, Mendelian genetics. There are some diseases like diabetes type 1 and type 2 that has some genetic component to it, but it's the inheritance is what we say it's multifactorial. There needs to be some environmental add-ons as well that can contribute to a child making it him or her more likely to have the disease. Now, DNA is found solely in the nucleus. DNA is basically the super long strands that we see all the time. We have two sets of 23 double strands of DNA. That's the same as two sets of 23 chromosomes. And again, one chromosome is the same as one double strand of DNA. The only difference is that a chromosome looks much more solid because the super long strands of DNA has been wound around proteins to make it be more efficiently stored. So then they don't require this super huge amount of room to store the super long chain of nucleic acids. We can wrap it around a protein, kind of similar to what we do when we are knitting, where we can kind of wrap any loose threads and put it into a ball so then it takes up less room. Now, DNA, long thread-like molecule, and again, 46 DNA molecules, which corresponds with the 46 chromosomes. They're in the nucleus in most human cells. The average human DNA length is about 2 inches long. That's actually pretty long. And now keep in mind that we have 23 DNA chromosomes that are completely different from each other. So if I compare chromosome number one to chromosome number two, they're going to code for completely different proteins. If I compare chromosome two to three, they completely code for different proteins. So we have 23 of those different chromosomes. We inherit two sets, one from each parent. Now, DNA and other nucleic acids are polymers of genetic material we call nucleotides. Nucleotides consist of a sugar, a phosphate group, and some kind of nitrogenous base that allows it to differentiate one nucleotide from another. A single nucleotide, and then here we can see it a little bit better. A nucleotide is a phosphate group that's in yellow. Sugar, which DNA is deoxyribonucleic acid. So sugar is deoxyribose, and then a base. Here in the pink, the base is a large double ring structure that we call adenine. Now, some of you have heard and seen videos where you have adenine always with thymine, cytosine always with guanine, right? So ATGC. So those are usually what we see with bases, that they're common twos in what we call complementary matching base pairs. Adenine on one side is always fine with thymine on either side, and guanine, adenine is always with thymine, and guanine is always with cytosine. So DNA is made out of these polymers, and a polymer is just replicating uh, kind of materials that are similar, right? So in terms of DNA, the nucleotide's a polymer. You have a phosphate group, a sugar group, deoxyribose, and then a base group. And the base is what allows us to have some differentiation, right? The base can be adenine, a big ring purine, or, right, it can be guanine, cytosine, or thymine. Now, we don't have just one genetic material, we have two. The second type of genetic material besides DNA is RNA. RNA is actually going to have, right, very similar structure in that it has a phosphate group, a sugar, and a base. What makes RNA different is that they're almost always found as single strands, and RNA has a different sugar, ribose, ribonucleic acid, RNA, and the actual bases are very similar. So in RNA and DNA, we have adenine and guanine, they're the same. In RNA and DNA, we'll also have cytosine. The big difference is that in RNA, we replace uracil with thymine. So just a bit different between those two kind of DNA and RNA. Now, usually I like to make the comparison. DNA is like the book, right? A big, huge book that tells you everything about yourself, right? RNA is what we do with the book. Do we read it? Maybe it's a cookbook, and now we need to bring all the recipes together, and now we can cook something. The actual structure that allows us to do things with that book found in DNA is the RNA. RNA is the functional portion of it. So DNA just codes for proteins. RNA is going to read it, it's going to find the appropriate amino acids that corresponds to it, and it's going to make proteins out of it. It's the actual doer, right? Now, DNA structure and function. We all know DNA is a double helical structure, right? And resembles a spiral staircase. Each side piece is a backbone of phosphate groups alternating with deoxyribose. Contains step-like connections between the backbones that are pairs of nitrogenous bases. And it's probably easier to show you this picture right here. So what we're talking about here with the sugar phosphate backbone is, take a look at the far left where we have the yellow, that is the phosphate group, and then the kind of the pentagon looking sugar, right? We call it a sugar phosphate backbone because it makes up the actual megastructure of DNA. So a lot of people kind of compare this to steps, right? So this is the actual structure for the steps. So the sugar phosphate backbone makes up the actual ribbons that make up our double helix. Now, the bases right here stick out of the middle. So adenine sticks in the middle, guanine underneath it, thymine underneath that, guanine underneath that. All right, they move, the bases face each other. So on the other side, if we have adenine on one side, the other side has to be complementary, thymine. And the reason why adenine is always found with thymine is simple. Adenine and thymine maximizes the number of hydrogen bonds. Guanine and cytosine maximizes the number of hydrogen bonds. And remember, hydrogen bonds are the only thing keeping these two sides, right? These two intricately linked double helical structures together. If the hydrogen bonds have been removed, those two strands of DNA separate from each other completely. So in order for us to actually make this double helical structure that we always see as DNA, well, in order for that to happen, adenine and thymine need to be close together so they can hydrogen bond very tightly. Guanine and cytosine, same thing, need to be close together and they maximize their hydrogen bonds. It is only when that happens do we actually allow the DNA molecule to become a double helix without falling apart. If we would say adenine is with cytosine, Well, when that happens, you don't maximize the hydrogen bonds. And what occurs then is the actual, right, bases, because they're no longer maximizing the hydrogen bonds, you don't have that interaction between the two sides anymore. And the two sides of the DNA molecules will fall apart. And no longer will it be double helix, no longer will it be actually DNA. Now, we call this where adenine is with thymine and guanine is with cytosine, the law of complementary base pairing. Now, it also tells us a couple things. If we know one side of the double helical chain, we know the other side. So if all we have was one side right here, adenine, guanine, thymine, guanine again. Well, the other side, we know with adenine has to be thymine. Guanine has to be cytosine. Thymine has to be adenine. Guanine again is going to be cytosine on the other side. How can we tell? Let's take a look. There it is, right? Why? Because we always see them together, always. So we see adenine on one side, always a thymine. Opposite is always true, right? If thymine is on one side, the other side is going to be adenine. Now, because of this, if we know a specific area of DNA has a specific number of adenines, we can tell the number of the other three bases. Meaning what? If adenine in one specific area, let's say we say it has 15% of that of the bases are adenine. That would tell us, number one, we know all the other three bases. How? Well, the four bases combined, those are the only four bases we have. So out of all of them, they'll make up 100% of the bases that's found in that area. I said there's 15% of that area, right, is going to be adenine. That means 15% thymine. Now that makes up 30%. Now, how do I know if 15% is adenine, 15% has to be thymine? Simple. Love complementary base pair. I see the same number of adenines as thymines, 15% each. Combined, when you add it up, that's 30%. That means that the other two bases, guanine and cytosine, make up the other 70%. And they're also found in equal numbers. So 70 divided by 2 is 35. So guanine and cytosine in that region will be 35% each. In other words, by knowing one of the number of the bases, you know all of them. Again, 50% adenine, that would mean 15% thymine. That's 30%. Guanine and cytosine are found in equal numbers. So 35% each. That is what allows us to understand this law of complementary base pairing. In terms of a gene, we know that DNA codes for genes, and that's all it does. It codes for certain genes, right? The genes code for specific proteins. Remember, in terms of protein, protein gives us function. So these are the enzymes. These are the hormones that we use in the body. They give cells in the body specific functions. So in terms of gene, a gene is any region of DNA that codes for a protein or multiple protein. We know we have hundreds of thousands of different proteins our body makes. So when we first started with kind of trying to decipher the genes that we have in all of us Homo sapiens, right? And it started in the mid 90s. We had strong enough computers that can actually go through the DNA. What we figured is we have hundreds of thousands of different proteins. We should have hundreds of thousands of different genes. And what we figured out was that we don't. We only have about 30,000 genes. That means that there are some genes that can code for more than one protein. But genes are nothing more than a sequence of DNA that codes for specific proteins that's specific for you. And everybody's genes are going to be slightly different than anybody else's, except for identical twins, right? Humans have about 20,000 genes, 30,000, only about 2% of total DNA. Other 98% of DNA is non-coding, meaning that only a small percentage of our DNA is going to be coding for specific proteins. Now, what does the other 98% mean? I mean, does it mean it's not important? No, not at all. All right. A lot of the 90% of the protein of the DNA is going to code for when to make the protein, how to make it, when to activate the actual gene so that you can make the protein. Where does the start and the stop end of that gene? So then you don't make the protein too long or too short. So all of these are regulations or regulator genes, right? That we find on DNA. So even though we say like 98% is non-coding, doesn't mean it's not important. It just means it doesn't code for specific proteins. It does in some or another help us kind of figure out how to make the protein. Where does where does that sequence begin for the protein I want to make? When do I want to make the protein? Because the last thing you want to do is make a protein when you don't need it or when you really don't want it. An example is what would happen if you are dieting, you haven't eaten, and your blood sugar is really low. What happened if your pancreas started to secrete insulin at that point, right? It's normally a protein hormone that our body needs. But what happens if you secrete it at the wrong time? Well, what happened is your blood glucose will drop off a cliff and you might die from hypoglycemia. So not only do we know how to make the protein, we need to know when to make it. And that's what some of these non-coding areas of DNA allows us to do. Now, chromatin and chromosome, what's the difference? And again, some of these are going to sound very similar. Chromatin, fine filamentous DNA material, complex with proteins. That's your chromosome, right? We have 46 chromosomes. We're going to have 46 chromatids and chromatins, right? Now, the thing is, once we start, we only start seeing the DNA, the chromosome, the chromatins, when we're about to divide. That's when we see it. Now, this is actually a good picture of just what chromatin and chromatids are and what a chromosome is, especially way down here, right? Whenever you see what looks like an X looking chromosome, this kind of looks like an X, right? Whenever you see that, that means that that chromosome has undergone replication already. We've duplicated the number of chromosomes. So there's actually two chromosomes here. But since each of the chromosomes are connected to each other, it only counts as one chromosome. Weird, right? Right. So you have one arm, right? And then right here's one arm, right here's a second arm. Since they're connected to the middle by the centromere, right? They're connected. It only counts as one chromosome. Later on, we're going to see that we're going to separate it. As soon as we separate it, it counts as two separate chromosomes, right? Keep that in mind that it's weird, but that's the way we kind of give it the arbitrary wording, right? When we have one chromosome by itself like that, right? We say it's one chromatin or one chromatid. And then another one, right? When it's together and it's connected, it's only one. There's genetic material for two, but since it's connected, it only counts as one. Once we separate it, even a micrometer, we just have a slight separation. Immediately, it counts as two separate chromosomes. But when they're connected by the centromere region, it counts as one. Again, when you see chromosomes and it has what looks like a double arm area connected in the middle by the centromere, right? That chromosome has undergone DNA replication. It has duplicated itself with a second arm. But we don't count it separately yet because they're connected to each other. It's only when we start separating it that we count it as two separate chromosomes. I know, it's kind of confusing. All right, now I kind of mentioned that chromosomes right here are basically DNA molecules that have been kind of rolled on proteins to make it a little bit more efficient to package so then it doesn't take up as much room. Well, the actual proteins that we use to wrap and roll the double strand of DNA is called a histone. Histone, right? Histones, proteins crucial for DNA packing. They cluster in groups of 8 molecules. DNA molecules wind around the cluster, kind of like how we wind a thread, right? Chromatin consists of thousands of repeating nucleosomes. So a chromatin are the double strands of DNA that are wrapped tightly around lots and lots and lots and lots of histone proteins. Right, so review those. Uh, each chromosome is packed into its own region of the nucleus, found in the chromosome territory. They're permeated with channels, allowing regulatory chemicals to have access to the genes. We need some way to control when we activate certain genes. You just ate, so now we need insulin to be released. We're going to have certain proteins go into the actual nucleus of our pancreatic cell and activate the gene for insulin. So then we can produce insulin in a huge amount, so then we can release it for the whole body to use. In non-dividing cells, the chromatin is not static. They change moment to moment according to genetic activity of the cell. In other words, there's always something getting turned on and something getting turned off. So if you are, in a given cell, you're always reproducing some kind of protein. So you'll always have certain genes being activated with other genes being inactivated. So then we can make the proper amount of the proteins we need. Right. Chromatin, and I'm trying to go through this, not super fast, but fast enough to kind of give you the gist of what we're covering here. And then not going too slow, so then it becomes overpowering, right? When coming back right here, in certain cells, genes get turned off, and in other cells, genes stay on. Now, what does that mean? That means it's crazy, but if we look at cells of the pancreas, we have cells right next to each other. One of them are called the alpha islet cells. Another one, it could be actually a few, like tenths of a millimeter next to each other. And that was, those cells can be called beta islet cells. So they're very close in proximity, yet they are going to be completely different in what they produce. The alpha islet cells produces glucagon, beta islet, insulin. So when you eat, we activate the beta islet cells and insulin is produced. Well, what happens when you are hungry? When you're hungry and you're fasting or starving, the last thing you want to do is produce insulin because your blood sugar is low. So instead, we need to release our stores of glucose so then the brain can use the glucose for itself. So you're fasting, you're in a diet, right? What happens? Your blood glucose has dropped because you haven't eaten because you're on a diet. So you need a steady amount for your brain to use. And since you're not eating, you're not getting that steady amount through the diet. The good news is from your last meal, you stored some of the glucose that you ate as glycogen. That glycogen can be broken down to glucose in the liver. So then we can free up that glucose. But that only happens if another hormone gets released. And that hormone is from the alpha islet cells found in the pancreas too, that is just a few millimeters away from the beta islet cells. Right? So we have two cells that are incredibly close together. Yet one cell produces insulin that will decrease your blood glucose, allowing the glucose to go into your cells. The other one, right, tenths of millimeters next to it. Well, that alpha islet cells produces a hormone that's completely the opposite, called glucagon. Glucagon will activate your glycogen and cause those glycogen to be broken down to glucose. So then you have a steady amount of blood glucose for the brain to use. So completely different hormones produced by cells that are millimeters or tenths of millimeters apart. How? Well, some of the genes are turned on in some cells, some of them are turned off in other cells. When preparing to divide, cell makes a copy of all nuclear DNA. Remember, we grow and we replicate from one fertilized egg that's got 23 chromosomes from dad, 23 from mom. We can't add another cell from the outside because then it won't match what we have already as a fertilized egg. So the only way we can grow into the person we are now, to being a fetus even, all right, the only way we can grow is by increasing the number of cells. And we do that by one cell growing, doubling everything, doubling the DNA, doubling all the proteins, and then dividing into two cells. Those two cells then undergo DNA replication, double all the organelles and proteins inside that cell, double the size of the cell, and then split into four cells, right? So the only way we can grow to become who we are, to the trillions of cells to become who we are, is to increase size, double everything, including doubling the chromosomes. When we do that, now we can make two identical daughter cells out of the one parent cell, right? So one of the big things is we have to double the amount of chromosomes, and we call that DNA replication, right? Each chromosome then consists of two parallel filaments of identical DNA called sister chromatids. They become visible with a light microscope, and each strand, each double strand, called a chromatid now, is going to be connected to each other at the central region called a centromere. So right here, this is one strand, one double strand. This is a second. When you see this X-looking double arm chromosome, right, it means that we have doubled the amount of the genetic material. But since it connected to each other, though, it only counts as one. What's crazy is that as soon as we separate it later on, it's going to count as two separate chromosomes. But when they're connected, it only counts as one. RNA structure and function. Remember, DNA is that master cookbook, right? That tells you how to make all the dishes that a chef or cook might make. So it's important because it's going to be there to give us the, the ingredients list and how to make things. RNA is going to be the one, it's the cook, it's a chef, is the one that reads it, right? It's the one that brings all the ingredients together. That's what RNA is. RNA, ribonucleic acid, smaller molecule, but resembles DNA, can have less than 100 and just over 10,000 bases per molecule. Now, remember, double-stranded DNA might have billions of bases. We're talking about 10,000 most. There are three types of RNA. There's only one type of DNA. The three types are messenger RNA, ribosomal RNA, and transfer RNA. All plays a huge role in how we make proteins. It's usually one nucleotide chain with a protein giving it a little bit of structure, right? It's got ribose instead of deoxyribose as a sugar. And in terms of the bases, we still see adenine, we still see cytosine, we still see guanine. The only thing we don't see is thymine, and it gets replaced with uracil, right? So we have adenine on one side as we try to read something. The other side, well, it should be thymine if it's DNA, but it's not DNA. It's RNA. So adenine is with uracil. Right. So let's talk a little bit about protein synthesis. Right. When a gene is activated, messenger RNA is made. So let's say I want to make insulin. Again, right? It can be found on one of my chromosomes. Do I need to replicate the whole chromosome? No. Usually a gene is just a specific area in that chromosome. So I need to know when it starts and when it ends, right? So I have, in terms of the gene itself, it's got an activation point, allowing our enzyme called RNA polymerase to basically clip on to that DNA gene. And again, the gene is just a sequence of bases, right? That tells you then which amino acids goes where. Again, we're making proteins. Proteins made out of amino acids. So messenger RNA is going to be produced once we start reading a gene. Messenger RNA is smaller, and it's just that gene that is specific for the protein I'm making. All right. You might have 100 other genes on that chromosome. I don't care, right? I don't need to make those proteins. I only need to make that protein that corresponds with insulin. The other 99 genes, I don't even bother. I only need that one gene to be active for insulin, right? So I have enzymes that will then bind to that specific region, a binding site for the gene, and start reading that gene and base pairing. That's all we do. As we base pair, we form messenger RNA out of it, and the messenger RNA is then transported out into the cytoplasm. The messenger RNA is complement to the gene. So on the gene, I might see adenine, while in messenger RNA, I put uracil in it. On the gene, I might see guanine. Then what complements guanine? Cytosine will be found on the messenger RNA. So all we do is read one side of the double-stranded gene. And as we read it, we basically transcribe it into messenger RNA, right? Now, DNA, this protein synthesis consists of uh, two, two specific mechanisms. The first one is called transcription. We take DNA gene and turn it into messenger RNA, a lot of it, right? Remember, we need enough insulin for the trillions of cells of our body, trillions of them. So we need to make it fast and we need to make a lot of it. So we have one gene. As soon as we read one, another one reads the other, and then we start making enough messenger RNA so that we can make enough insulin in enough quantity. So once we make messenger RNA, right, we take messenger RNA, take it into the cytoplasm, bind it with ribosomes, and we start basically finding the correct amino acids for it, right? mRNA codes for proteins, and it occurs in the cytoplasm. I don't want to go into too much detail on here, but just understand and that, you know, messenger RNA, that's like in my 201 class where we have a little bit more time to go in detail, I talk about, like messenger RNA being a photocopy, right? Imagine you're making your famous lasagna, right? And it's basically from a cookbook, or it's a recipe from your parents, and they got it from their parents, right? And they got it from their great grandparents. So this recipe book is pretty old, and you want to make a lot of that lasagna because you're hosting a party. Well, if you're hosting a party, you might only have maybe one oven, so you might have somebody that helps you by making lasagna in their house. So you don't want to give them the actual cookbook because it's fragile and they might lose it. So instead, what do? You give them a photocopy of it, a photocopy of that recipe. So then if it gets lost, it doesn't matter. It's just a photocopy. You have the original copy at home, right? That's the thing about messenger RNA. Messenger RNA, we can make a lot of it and we can transport it out. If it gets lost, it doesn't matter. All right, the gene is safe in the nucleus, tucked away. If I need more messenger RNA, right? If I need more of the photocopies, I can make it. Right. So let's take a look at the cell life cycle. So this is the cell life cycle right here. There's a G1 phase, an S, a G2 phase, and then mitotic phases. Remember, all of our cells come from previous other cells. So one cell becomes two. So we have a fertilized egg. That's the very beginning. Then it'll undergo DNA replication. It will increase the number of all the organelles, increase the number of phospholipids. So then it's got enough plasma membrane to form two cells. Once everything has replicated, including the DNA, now we're going to split the cell in half. So then by the time we're done, the two cells we're producing at the end is going to be identical to the original one cell that we started with, right? In order to do that, we need to double everything. So the very first thing, so imagine I have that fertilized egg, right? One half comes from the sperm, half the DNA comes from the egg. They fuse to form one whole cell, a zygote, right? Now we somehow have to increase in size and then divide. We need to increase the number of DNA and then divide. So then we have two cells instead of one. Then each of those two cells undergo the same mechanism. They double everything, double the organelles, double the cytoplasm, double the phospholipids, and then those two cells becomes four. Those four cells becomes eight, and then 16, right? And then you grow in a very linear fashion. Now, let's talk about how we do this. G1 is called the first gap phase, right? Interval between well, from the time the cell is first born, right? The cell then carries out the normal task of a normal cell, is going to increase the number of DNA nucleotides, RNA nucleotides, right? So that it can get ready for the next phase. Now, keep in mind, this is one big circle. If you look at the green arrow starts at where the orange arrow ends. It goes all the way around, right? G1, S, and G2 are all part of the green arrow area. We call the green arrow the interphase. This is where we kind of get everything ready so then we can have cell division and form two cells out of one. So G1 gets everything ready for the next phase, which is called the S phase, right? The S phase right here is where DNA replication occurs. Remember, we need to duplicate the number of chromosomes. We're producing two identical cells. So we only started with 46 chromosomes. If we split it, then we have 23 chromosomes. That's not good, right? Now we have 23 chromosomes in one cell, 23 in the other. It doesn't match. So what we need to do is double the chromosome count for that cell that will divide. Double it during the S phase. So that cell, 46 chromosome zygote, will go into the S phase. Then it's going to undergo DNA replication. It's going to have twice the number of chromosomes at that point. And then we get ready for it to split. Once we have DNA replication in the S phase, now we can get ready for cell division. And that's what happens in the G2 phase. G2 phases, right? We've already replicated the chromosomes. Now we get ready to split everything in half, right? So all the mechanisms there to split everything in half, well, they're all produced in that G2 phase, right? By the end of G2, we've doubled everything. We've got all the machinery ready to split things in half. And now we hit what's called a mitotic phase. Now, there is a mnemonic for this, and it's called IPAY-MATC. I for interphase, pay, prophase, M, metaphase, A, anaphase, T, telophase, C, cytokinesis. Again, mnemonic is IPAY-MATC, right? Now, let's talk about this mechanism of mitosis. Mitosis is cell division that produces two genetically identical cells that we call daughter cells. That one cell that started the process is called the parent cell. So by the time we're done with mitosis, we produce two cells that we call daughter cells. Those two cells should be identical to each other and should be identical to the original parent cell. The main function of mitosis is a lot, right? This is how we get from one fertilized egg to the trillions and trillions of cells that makes up who we are. We have so many cells we can't even agree to the nearest trillions. And all those cells are formed via mitosis. Mitosis allows us to grow, right? Allows us to replace cells after they die. The best example of this is what happens when you have a sunburn, right? Sunburn causes acceleration of healing, and cells from underneath replace the old ones that died off from being burnt. They are, right? So repair of damaged tissue, replacement of cells that die, growth, right? All functions of mitosis. Every cell that has a nucleus can undergo mitosis. So the red blood cells, no nucleus, they're the only ones that do not have the capability to undergo mitosis. They have no genetic material. Again, there are four phases: prophase, metaphase, anaphase, telophase, right? Prophase is where we start. We've already doubled everything. We've already doubled the DNA. Now we're ready to split. Prophase is when genetic material becomes easily seen, right? The genetic material compacts into chromosomes. They're easier to distribute to daughter cells than chromatin, right? Nuclear envelope disintegrates. Centrioles sprout spindle fibers. Remember, these are there to kind of move the spindle fibers, and the centrioles and microtubules are there to kind of move your chromosomes around so that it can split in half. So in meiosis, in a prophase, we start seeing it. The chromosomes, the nuclear envelope starts to deteriorate, and now these spindle fibers come out that will eventually connect to the machinery in the central area of the chromosomes. So then it can move everything around. So then we can split everything in half. In metaphase, what happens is, here's a good picture of metaphase. Notice how all the chromosomes are lined up right in the middle. That's what happens in metaphase. In metaphase, all of the spindle fibers sprout out. They connect to the middle, called the centrosome, right? And they have specific mechanisms in the centrosome called kinetochores that kind of allows you to attach and move the chromosomes to the middle of the cell. So metaphase starts with M, middle, or midline, right? This is where, in metaphase, we see all the chromosomes move towards the middle so that we can split everything in half, right? In anaphase, since all of those chromosomes are lined up right in the middle, what we're going to do in anaphase is split those chromosomes in half. So then each eventual cell will get one copy of one chromosome from one parent. So you get one chromosome number one, and it goes to one cell. The other chromosome number one goes to another cell, right? So then this is the way that we should see it in all of our cells. Anaphase enzyme cleaves the two sister chromatids apart at the centromere. Single-stranded daughter chromosomes migrate to each pole as motor proteins in the kinetochores crawl along the spindle fibers. Basically, the chromosomes are moved away from each other and move towards the ends of the cells. In telophase, chromosomes cluster on each side of the cell. Rough ER makes new nuclear envelope around each cluster. So here is the anaphase where we separate the chromosomes right here. They color code it so that we can tell that we have one chromosome one on one side, right? The other chromosome copy that's identical goes to the other side, right? Now, once we separate all the chromosomes, we hit the telophase where all the chromosomes have migrated to distinct areas that will become separate cells. At that point, nuclear envelope redevelops, and now we can completely pinch off in cytokinesis. Right? So take a look at these, right? I know I'm kind of quickly going through this. Read the work, all right? Read the book if you have any questions.